Thanks for joining us today. Again, Amy's great introduction to the webinar today is going to be on viability using Hoechst PI reagents on Celigo. This is a very common and easy to use assay if you're just getting started on the Celigo and you want to try and do some fluorescent assays. This viability assay will go a long way. I'm going to go through the process in a manner that's going to show you how to do something if you've never done it before, but then you can come in and add drugs at any time after you've seeded your cells. And so I just want to share with you, this is going to be just with your cells and the dye, no drug. So I'm going to talk about how to plate the cells and how to stain them. So the easy method of those two things, and then you can add drug if you want to or not. And then I'm going to show you how to set up the Celigo for how to acquire the images on the Hoechst and the PI fluorescent stained imaging, and then how to analyze the images to detect those objects. And then we're going to show you how to review the data and check out the images in the results tab. And then I want to just kind of show you one last layer is putting data into graphs. That's always the part that kind of sometimes gets missed out, but it's important to kind of bring your cell data into a completion. So I'll show you how to do that. So let's get started, and we'll start showing you the process to plating your cells first. So in the lab, you're going to need some cells that are growing well, proliferating. So when you're having your cells in the flasks that are adherent, you want to look at them in the microscope and see that they're about 90% confluent, 95% confluent and proliferating well. You're going to remove the PBS or remove the media and wash it a couple times with PBS. And then you're going to come in and add trypsin. This will digest the attachment factors of the cells and lift them off the flask. You're going to incubate this trypsin for about 5 minutes in the incubator. And then you'll see the cells kind of floating. You're going to stop the reaction by adding 4 mLs of warm media. The FBS stops the reaction of trypsin. And then you want to pipette it up and down until you get the cells in single cell suspension. Then you're going to count the cells. I'm going to slow this down a little bit, I'm talking too fast. And then you're going to pipette the cells into a single cell suspension, and you want to plate them to be serial diluted in a plate. So this is the plate map for today's experiment. So we're going to do 40,000 cells in column A1, and then serial diluted all the way over to column 12. So how do you prepare the serial dilution? So you're going to have one tube. You're going to make the cell suspension a concentration of 400 cells per microliter in 2 mLs. Then you're going to set up 11 more tubes with 1 mL media. Then you're going to pipette 1 mL from this first tube with 400 cells per microliter into tube which has 1 mL. This is a dilution factor of 2, so now you're having a concentration of 200 cells per microliter. Continue this serial dilution of 1 mL into the next tube and until you get to the last tube. Now you're going to pipette 100 microliters from these tubes into each of the wells in the 96 well plate according to this plate map. So column 1 is tube 1, column 2 is tube 2, and so forth. Then I like to centrifuge the plate just gently at 100 or 1,000 RPM up to speed and then turn it off to settle the cells. If you don't have a centrifuge, just leave the plate in the hood for 10 minutes to let them fall down naturally. And then put them in the incubator overnight so they can adhere to the bottom of the plate. Alternatively, you can do the serial dilution in the plate if you're comfortable with a multi-channel pipette to do the serial dilution. Next, we're going to talk about the staining of the cells after they have adhered overnight. At this point, if you had drug, you could put it in and let it incubate on the cells for how many hours, 24 hours, whatever, and then do staining the following 24 hours. So this is the staining materials. We're using Hoechst with a stock concentration of 20 millimolar, and then propydium iodide is 1 mg per ml. We'll need our cell culture media. You can use PBS if you prefer, but some cell types don't like PBS. And then the 15 ml conical tube, and then your pipettes and a multi-channel pipette and a reservoir. Okay, so for the cell staining, we're going to first create a master mix that has a 2x concentration of our final stains. So what does that consist of? So in a 15 ml conical tube, you're going to add 10 mLs of the cell culture media, and then 8 microliters of the Hoechst, 40 microliters of the PI. You're going to cap this solution and mix it. Pour this mixture into the reservoir and then pipette 100 microliters into the wells on the plate containing the cells. You can use this multi-channel to do this. It's a lot easier if you have like an 8-channel or 12-channel multi-channel pipette. And then you want to be gentle when you put that in there so you don't disturb any dead cells. This becomes a 1x staining solution. You'll want to incubate this in the incubator for about 30 minutes and then image on the Celigo. The final 1x concentration of these dyes is 8 micromolar Hoechst and 2 microgram per ml per PI. An alternative method, if you had put drug into the plate, your volume's probably around 200 microliters. You could come in with a 5x staining mix solution and add 50 microliters to the well. That's another alternative method. So now we'll move over to the Celigo software and we'll show you how to acquire the images and how to analyze and review the data. So in the Celigo software, we're on the Start tab. You can see the workflow in the upper left-hand corner. And we're going to click this Create a New Scan. And now we need to tell the system what we're going to image today. So we're going to look at a 96 well plate format. So these are all the different well formats that the system can image. And then we're going to tell the system what manufacturer and vendor catalog number we're going to image today. This is a list. It's alphabetical from the manufacturer and the catalog number. Today we're looking at the Griner 655090. It's a black wall, clear bottom plate. Then we're going to give our plate a unique ID name. Sometimes I'll just put the date with my initials and something unique to the experiment that I'm going to do that day. But today's training is, I'm just going to put down generically Viability Hoechst PI. And then you're going to click the word Load Plate. This will move the stage on the system out of the front of the Celigo. You'll put your plate onto the stage, and then you're going to click the OK button. The stage will move back into the Celigo and move over to A1, the well A1. And the software now migrates into the Scan tab, and we can start setting up the parameters here. So first we have to tell the system what kind of application do we want to use today. We're going to select the one that's called Cell Viability Dead plus Total. So these suites are all listed alphabetically, so we're going to go down to Cell Viability and then see the first one, it's Dead plus Total. It's a 2-channel application. We're going to look at the red and the blue fluorescence. All right, so first step, I like to look at my total channel first because it has stain in every cell, whereas the dead I might not have that many cells to look at to set up my focusing and so forth. So I switch over to the total channel first. And then the default settings always come up with blue as your illumination, which is going to work for our total Hoechst stain. And the default illumination is at 10,000. It's on the dimmer side for this stain, so I know it's hard to see this on the screen, but I just want to click snap and see, do I have some staining? It's very hard to see probably on this video, but the cells are there and they're stained. It's a little dim and a little out of focus, so we're going to fix that. So let's move over to a more central well in the plate. I tend to gravitate towards C3 more than other wells. I avoid setting up the focus on the outer wells of the plate. All right, so you can kind of see, it's probably a little hard to see with the black screen on a video, but there are cells there slightly dim. So let's first click Focus Setup and get these cells in focus. We're going to use the hardware focus mechanism to do the focusing for the acquisition, but we need to tell the system what to focus on. So click Register Auto. This now engages the stage to move up and down, and the system's going to try and figure out where the cells are in focus. I know it's hard to see, it's a little dim. And then once it's done, it'll say that it's registered on C3 in the blue channel. See, there it is. Now we can close this dialog box by clicking Focus Setup. Now we're going to adjust the exposure. You're like, really, are there cells there? Yes, they are. So what we want to do is find our objects and hover the mouse over the objects and get the pixel intensity to be in the middle of the camera range. So we can just turn up the exposure just a little bit and click snap, and you're going to see the cells appear. So now the camera range is from 0 to 255, and we want our objects to be in the middle of that. So that's between 100 and 150. So these are Hoechst stain cells, which go on the nuclei, and you want to have your majority of your cells to be in this camera range. You're going to have a few cells that are going through the mitosis, so they're going to be super bright. You don't want to focus on those. You want to focus on the flat one, which is the majority of your cells. So just keep turning up the exposure. I go up in groups of 10,000 until I kind of see that I've met where the cell pixel intensity is in that middle of the camera range. This is a good general rule of thumb for all your fluorescent dyes. To get your object in the middle of this camera range is good, because then the analysis settings are going to work more easily in the default settings that show up on the screen. So, and also the background. You want to make sure your background of the dye stays below 50. This dye has got little background, not a problem, but there are other dyes out there that have some inherent background. You ideally don't, the more exposure you turn up, the more background you might get, so it doesn't help. You might want to dilute it out with some PBS or something if you have that problem. All right, so this looks good. We've set up the focus and illumination and exposure for the blue channel, or the total channel. It looks good. Now we can go over to the dead channel and set up the focus and illumination for these, for this type of imaging. So we'll change the illumination to the red excitation and emission filters, and then we'll click snap. And the default setting of the exposure is at 10,000, so let's turn that up a little bit and then click snap. I prefer to use snap over live, because if you have a fluorescent dye that actually is sensitive or photo bleaches, meaning you take more fluorescent energy away from it the more camera time you have on there, you don't want to have that happen. So I just use snap instead of a live camera for fluorescent imaging. Now we want to get our cells in focus for the blue, or for the red channel, and I use the word find focus. On the other channel we use the focus setup, because that's where we tell the system to register for the hardware focus. Now we want to figure out where the red cells are in focus, so we do find focus. It's moving the stage up and down and trying to see where the fluorescent dead cells are focused. And it says it's 12 microns negative from the blue channel. And so that's how we want to acquire these red images, is 12 microns below the blue channel. So we click set offset, and that populates the number up here at the top. So the system is now being told to acquire the red channel images at 12 microns below the blue channel. Now that they're in focus, we can set the object pixels to get them in the middle of the camera range. So let's turn up the exposure. 30,000 is not quite enough. So the recipe for the Hoechst PI that I gave in the previous screen, if you follow those and your cell types are similar, then basically the exposure settings that I'm setting today for the blue and the red channel will probably be good for you. So you don't have to start at 10,000. You can probably go directly to, like, for the blue channel, you can probably go directly to 70 or 80,000. And for the dead channel, depending on how dead your cells are, how quickly the dye goes in, the PI, you can go between 30 and 50,000. All right, so we've got the illumination and focus offset set for the dead channel. The last thing we need to do here is select the wells that we want to acquire. So click the selection button and then highlight the wells by clicking this space between the A and the 1. That pretty much highlights all of the entire plate. And then click Start Scan. The system moves over to the Analyze tab and starts to acquire the images according to those settings that we just provided. And when the system has saved the image to the computer, the wells on this plate maps turn green. And you can also see the progress bar down below is also telling you how much of the acquisition is saved or acquired. While the system is continuing to do that, we can start to look at the images that have turned green. So click on the A2 well, and you can see that the image for that well is displayed on the screen. So we have our Hoechst stain, that's pretty obvious there now, and a few dead cells, not many. So what I want to do now is set up the graphic overlay to correctly identify the cells. So we'll turn off the dead channel image, so turn off image display for dead, and turn on the total graphic overlay. So the yellow outline is currently looking at the blue Hoechst stain and identifying those total Hoechst stain cells. On the left-hand column, we're going to start selecting the parameters. So I do use the well mask. You don't have to, you can, but it eliminates the excess of objects outside the well area from being detected. And then there's the channel identification. We'll switch it over to total. And these default settings, again, if you chose a good pixel intensity on the scan tab, the default settings are pretty good starting point. Since this is a nuclei stain, they might be smaller than 10 micron. It will be cell line dependent. And also check this little auto analyze button. Basically, anytime you make a parameter adjustment, it'll automatically update the graphic overlay as you hit enter to the system and the graphic overlay. All right, so we're just moving around the image to see if we find some other features that we want to correct the parameters on. So these clusters are not being separated. So let's use separate touching objects to split up this cluster of cells. All right, so that's pretty good. Now we can change the channel over to the dead channel and then also turn on the image for dead, turn off the blue total channel and turn on the graphic overlay for dead and turn off the total graphic overlay. So basically we're only looking at the dead image and graphic overlay. So the default settings for this look pretty good. It's a starting point. We may need to adjust the intensity threshold and the diameter. Remember, our images are 1 micron per pixel, so if you see the cell diameter, it says pixel, so think of 10 microns. When you see the number 10, it's 10 microns. So a cell is about 10 microns. You may need to, if your cells are dying, they kind of have this ballooned, bloated look about them. That's why you see these flat, round discs, if you want to say of PI. It's pretty much the cell is really kind of exploding. I don't know. And they're dim. So what we'll do is turn down the intensity threshold because it's not super bright in the PI stain. And you can see that I was able to pick up those dim PI stained cells. And I'm just moving around and turning up the intensity threshold. Another alternative, instead of bringing the threshold down, you can increase the diameter of that object detection. So you basically can move that up a little bit if you want to pick these rounded up dead cells. So here, what you do is you hover your mouse over the object and you look at the X and Y position to kind of see how big is your cell. These are in microns. So if you move the mouse from left to right, you'll see what the size of this object is, and that'll help you set up what the diameter should be. So we're about between 15 and maybe 12 and 15 pixels across this object. And so if you pump that number in there instead of 10, you're going to actually see the objects being detected as 1 instead of 2. There. So that's how you identify objects that are bigger, but you don't know exactly what size to put in there cell diameter wise, so that'll help you. So now if we move that intensity threshold back up to 4, which was the default, we still can pick up those flat, round, dim, dead cells, but we don't pick up any small, too much little debris. If you do have a little bit of debris cells that you know are not dead, it's just debris from PI maybe, I don't know. You can use this filtering out section. So currently the default is set at 10, and that's pixels squared. So if you increase that up, you can see the small little objects being removed. So I would say probably for dead cells, you could go 25, 30, or 40. It depends on your image. You'd have to kind of play that on your image to see. So now you can see both graphic overlays and images showing where the nuclei are identified as well as a dead cell, and they usually sit on top of each other, because if it's stained with Hoechst and it's stained with PI, it's generally in the same location. So if they're not touching or overlapping, the blue dead objects are actually excluded. It's not considered a dead cell, it's considered debris. So that's going to be excluded in the data. So you're really only getting dead cells. So that looks pretty good. So if you like these settings and you're like, oh, I want to do this experiment on a different plate with drugs next time, you can save these analysis settings for another time, another plate scan. So just click on that little disk save button up top and give it a name that's unique to your assay, and then just click OK. So next time you can recall these settings and not have to go through all the analysis setting parameter setup. Now you select the wells for analysis. We're going to do the whole plate. So we click the space between A and 1 and highlight the whole plate. And the system will continue to acquire the images and analyze them at the same time. So basically you're performing the acquisition while we're setting up the analysis parameters, and then you hit start analysis and everything's done simultaneously, and we can start to review the data. So here on the left-hand side, you can see the parameters that can be displayed. Currently, we're looking at the % dead. And remember our plate map, we have 40,000 cells on the first column 1, and then we're serially diluted. And you can see that I have consistent dead cell population regardless of the cell number that I put in there. So this is kind of more of an exercise to see what cell number should you put in your well. I would probably not go above 40,000. Your cells might overlap in the well and you won't be able to clearly separate them with the Hoechst stain. And you can also see how low you can go to the point where you start to see that maybe it's not a good number to go below if you're going to do a drug screen study. I would probably, and also you want to know how many days do you need to have your cells in there for the drug treatment, and then allow for the proliferation to not exceed 40,000. So here's our dead count for the population and then the total. So even though I plated 40,000, they had some division overnight, and now I'm a little bit above 40,000. And the heat map is the little eye icon sitting next to the word heat map. Allows you to color code your cell or well data according to the numbers. And I think it's the top 5% and lowest 5% that defaultly come up the green and red, but you can automatically change these or manually change these values to whatever display you want. So if we put in our parameters, you can see where we've color-coded them according to our values that we want here. And then everything in between is yellow and below is green. And you can change the colors. If you don't like the red or the green, you can change the colors. Okay, so let's click on a well and you can see the high resolution image in the display area. So here we have the Hoechst stain. And as I scroll in, I can see the cells, and the graphic overlay will appear when you get to a certain zoomed in level. I think it's when the image resolution is back to the 1 micron. And now you can see any dead cell has to be touching a nuclei to be considered dead. So you can see that is currently happening. That's correct. And so we've definitely called out our dead cells with the blue graphic overlay. That looks pretty good. And you can see the parameters, or excuse me, the results for this well are displayed on the left-hand side. And besides the percentage, you get the counts. And then also you can get mean intensities, integrated intensities for both the dead and the total channels. Excuse me. So let's export this data into a CSV file. So clicking on this well level export will provide you a dialog box where you can save it, and it's a CSV file, so comma separated value. So when you open this in Excel, you're going to want to save it as a Excel file. All right, so we've looked at the images. Now let's go look at the data, and we'll put it into some kind of graphic format. Okay, so here we have a template that I created for this plate layout. So I know I have 40,000 as my highest concentration, and it's serial diluted to the right. And then I've already created locations for the data I want to present, so that's percent dead and total counts. And now we're just going to put the data from the CSV file into this page on the left of the tab. So we're going to open up my CSV file, and we're going to here's what the data looks like. The top half is your plate information, like your plate ID, the time you were imaging it, who was the imager, and then the data below that is the percent dead, dead count, total count, all the stuff that you saw on the Celigo results is now displayed here in the CSV file in a plate layout format. So typically you're going to probably be interested in the percent dead, and then you have the dead counts and the total counts. And also I just want to point out too, at the very bottom of this page, it also tells you the acquisition and analysis settings. So if you're like, well, how did I acquire and how did I analyze those images? So down here, it has all the information of the analysis for this data and image set. So I know that some of the terminology is not exactly translated word for word on the screen, but you can see the values are there. And then the illumination is down at the bottom. So we use 50,000 and 70,000 for the red and the blue. So that might be your starting point next time you do this assay. Okay, so let's copy this data set over to my template and paste it so we can see. All right, so we're copying it. So you can do Control A or just highlight everything. And then let's move over to the template. And the left tab is actually my location where I want to paste my CSV file information. So I just go to A1 and Control Paste B. And it looks the same, everything's good. And then it should automatically populate into my template. So now you can see I have the averages and standard deviations for the percent dead and the total dead counts and the total counts. So that looks pretty good. And then over on the left or on the right side here, I'm plotting the actual counts using the average total counts for my x-axis, and then I'm giving my percent dead on the y-axis for this graph. So you can see at the higher cell or wells with cells, the average is around 2%. And down here on the total count graph, I'm actually plotting my expected against my actual and see what kind of linearity I get for my cell plating and my Celigo analysis. So that's pretty good, not bad. So that is also showing you your actual up at there at the top. So this is just a good exercise. If you're just starting on the Celigo and you're trying to get familiar with your assay, you don't have to use any drugs. You just do this cell density plating to kind of get comfortable with the settings and the parameters and so forth, and understand how many cells in a well you should put. You can see, even looking at the images, you're probably at the top end if you're at the above 45,000 cells per well for a 96 well plate. It also is cell type dependent. So take that into account. If you're going to do some drug studies and your cells need to proliferate for 24 hours, you want to actually divide that in half or even maybe slightly less than that to allow yourself to not get to that maximum cell number per well after a 24-hour proliferation or drug treatment. You want to make sure your controls are not over confluent. So that looks pretty good. The kind of correlation about 99%, yeah, R squared, 99. Pretty good. Okay, so we've covered quite a bit today, and it's really a good starting experience for you on Celigo if you've never used the fluorescent stains on imaging cells before. This is a good assay to try, and it's also kind of a go-to assay when people are doing projects they're just getting started on. All right, so now we can open up the forum for questions. If anybody has any questions, we can have you type them into the webinar format, and then Amy can ask it for me.
Thanks, Sarah. I think that was a really great presentation. Yep, as Sarah said, we are taking questions, so feel free to type them into the questions bar, and me and Sarah can answer those for you. And while we're waiting, I would just like all of our attendees to know that we do encourage you guys to submit any topics for webinars that you'd like to see in the future. So if there's a specific application or assay or any topic for the Celigo or any of our other products that you're interested in hearing about and learning about, feel free to send them in. You can contact us by support@nextlim.com. We'd be happy to take your suggestions into account, and yeah, so just type in those questions for us, and we'll get to those in just a minute.
Amy, thanks for that plug, because I'm always looking for new, fresh topics to help people out on the instrument. In addition to that, if you guys have other types of dyes that you guys are interested in, you're like, oh, can that be done on Celigo? Just email that kind of question to us at support@nextlim.com. And if we haven't tried it before, what we can do is actually try it here in-house and see if it does work. So if you have a fluorescent dye that you're curious about, definitely contact us about that too.
Yeah, that's great. So I'm not really seeing any questions come through. So I guess that will conclude our webinar for today. But if any of our attendees, if you guys, you do think of a question later on, feel free to give us a call, or you can email us at support@nextlim.com. We'd be happy to answer any of your questions you guys have. Again, today the session was recorded, so you will receive a link to the recording later on. And you can watch it again, or you can pass it along to a friend or a colleague if you wish to do so. I hope you guys join us next month for our next Celigo user training webinar. Again, Sarah, thank you for a really great presentation, and I hope everyone has a great day.
Thanks, Amy. Thanks, everyone, for attending.
Goodbye.
Bye.